finally, we're going to be looking now at the synapse and what happens in terms of the chemical transmission. So remember we said that neurons operate through electrochemical signaling. So here's the chemical piece. We're looking at what happens at the level of the synapse. So we're going to start with this. So we already know about action potentials, and we know that an action potential is basically a change in voltage that causes a current of electrical impulse to move along an axon. But once it gets down to the terminal, then we have some other activities. So if you remember, the first thing that happens in response to that last piece of the axon going through its voltage changing, right, through the whole depolarization, repolarization, is that it then triggers that voltage gated calcium channel. And then calcium is able to enter into, here's the axon terminal, and it's going to activate these vesicles that contain neurotransmitter. So calcium is really important. Very low concentration in neurons, actually fairly low concentration out, but is a huge activator once it comes into a neuron or into a cell. So once those vesicles have been activated, they're going to then be moved down to the end of the terminal where we see the plasma membrane. They're going to fuse with that because they are little membranes themselves, or they're little bubbles, and they are going to fuse with that phospholipid bilayer of this membrane, and that's going to cause those vesicles to release their contents. The contents are going to exocytose out into the synapse, and there we go. And they're going to cross the synapse. So you can see that right here. Now, the synaptic cleft is really that gap. It's that little gap that exists between some presynaptic terminal and the postsynaptic cell, usually a dendritic spine, or it can even be on the actual soma of that neuron. But this is the space that electricity can't cross. And so now we're going to convert it to an electrical signal. I'm sorry, a chemical signal. And in this case, in a lot of these pictures that we're about to see, we're going to be looking at this in particular. We're going to look at acetylcholine. And acetylcholine, remember, it's ACH is its abbreviation. It's just its chemical structure. And so acetylcholine we find in the neuromuscular junction. We also find it in a lot of areas, particularly in the frontal lobe and in the cortex. So, but we're just going to take a look at it this way. So, understanding that we're releasing some of this neurotransmitter, in this case we'll say acetylcholine, you can see it right here. It's now going to move out and across the little gap, the synaptic gap, and it's going to activate these acetylcholine receptors that are on the postsynaptic cell. If this could be a muscle cell as well as it could be another neuron, depends on where we are. And so what happens is that's going to cause sodium, an influx of sodium. You can see the potassium is going out. So that's an efflux of potassium and an influx of sodium. And we're going to get some kind of a graded potential. We're going to get those little wavelets that you remember, and those graded potentials, if they sum to threshold, then we're going to get an action potential. Here you can see a voltage-gated sodium channel right here. Let's see here. Let's go to here. Whoops, actually, let me go back up here. No, I think I'll go, sorry. So let's look at this example of the neuromuscular junction. We have acetylcholine. It goes across the cleft, and it's going to allow, once it's formed synapses. Notice these look very, very different than what we looked at before. But Mia presented this when she presented acetylcholine. So here at the neuromuscular junction, this neuron, which is a motor neuron, notice it's all myelinated here, it's going to have, the synapses are right in here where those muscle cells or muscle fibers are. So when sodium and potassium go in, they cause excitatory postsynaptic potentials. And that voltage change, if it's significant, is going to cause the response in the muscle. Remember that in a lot of places in the body, there's different types of receptors. There are those which we call ionotropic, which are the very fast ligand-gated channels, very open quickly and close quickly. That is because acetylcholine was the first neurotransmitter really researched and understood. This particular type of receptor that's specific to acetylcholine, that is just a fast receptor, was called nicotinic. Because remember, they actually used nicotine to see what the effects would be on the receptor. The other type is the metabotropic. These are ones that are linked to other things within the body of the cell. Here you can see these G protein that are associated or coupled with the receptor. And so when the stimulus, such as whatever ligand we're talking about, if it's acetylcholine, if there's a receptor here, it's going to activate this G protein, which is going to activate a whole series of other things. So these were, when they were discovered, responded to muscarine, which is a compound found in mushrooms. So they're often also called muscarinic receptors, but slower receptors linked to these things we call second messenger systems. So that's that type. Remember that once whatever ions come in, they're going to come in and they're going to cause this little wavelet. Now this is showing in this graph, you see the EPSPs. So we have these excitatory postsynaptic potentials, just little waves, but they're still below threshold. They don't last long. They actually, they just sort of fade away, kind of like ripples on a pond. When they do that, at that point, there's really no change too much of the polarization of the cell unless there's enough of them. And when you sum those, when those are all summed up by the axon hillock, then we're going to see, if it reaches threshold, then we're going to see the action potential. So up here in this little picture, you see that there's several points of input here. These are excitatory synapses. And so there's little EPSPs being generated here, and then the axon hillock gauges that. Have we hit threshold? If we have, then let's open the first sodium voltage gate. So once we've let those neurotransmitters, whatever they are, out into the synaptic cleft, we have to do something. We can't leave them there. They go across, they activate receptors on the postsynaptic side, but they don't stay there, so then they're back out of here in this synaptic area, and they need to be quickly cleaned away so that the next signal that this neuron gets is able to respond accordingly. So so several ways we know we can clean up the synapse, and this one happens to be a mechanism that serotonin has, and it has a reuptake mechanism right here. So once this serotonin has activated this postsynaptic cell, it's going to get pulled in and then brought back into vesicles. So we can block that. That's what this little thing is here, using things which are called serotonin, selective serotonin reuptake inhibitors, or SSRIs. So that was one thing that we talked briefly about. So that's the reuptake mechanism. The other way they can be broken down is just by enzymes that degrade them. They break it down out here. Some of them diffuse away, just simply float away. And in some cases, astrocytes are involved. If you remember, we looked at GABA and glutamate in particular. Astrocytes sometimes act as middlemen, and they pick up the GABA, or in really most cases the glutamate, because glutamate actually can be converted into GABA, but they pick it up and do some chemical reactions that change it back, and then it can get back into the neurons themselves. So we didn't get into great detail on that, but just that's one mechanism to clean up, is to have astrocytes doing some of that. We talked a lot about neurotransmitters. I've done the videos on these. We know that there are some major ones that we've talked about, the amino acid ones like glutamate and GABA and glycine, simple amino acids, but these are very powerful ones in the nervous system. The most excitatory ones and the most inhibitory one that we know of are these ones right here. And then we have all these others, all these amines. Remember, these are just chemically, they have something called an amine group on them, so they have a nitrogen component, and that includes acetylcholine, which happens to have its own kind of category too, and then these three here, often called catecholamines, and that's dopamine, norepinephrine, and serotonin, and then a whole variety of peptides and neuropeptides that we looked at and that you all presented in your research presentations. I presented a little bit on nitric oxide and its role as this retrograde neurotransmitter, this gas that actually goes in the wrong direction. And then we also had a presentation from Bridget, I think, on, it was Bridget, on adenosine, and how adenosine, which builds up in the nervous system, in the brain, during the day because of intense use of cellular activity, also can act on receptors and helps to facilitate that feeling of sleepiness that we get. And so those are this is just a kind of to remind you of some of the things we already talked about. So finally, we remind ourselves a little bit of what we mean by excitatory. Some of these neurotransmitters will cause the excitatory postsynaptic potentials, so they're going to open channels that bring in positively charged ions into the postsynaptic cell. Some will be inhibitory, which are going to bring in ions like chloride into the postsynaptic cell, which will cause inhibitory postsynaptic potentials. And some are modulatory, meaning that they're going to change the activity of that neuron in some respect. So plasticity is what happens at the synaptic level that it means change. It means that the neurons are changing in some way. They're physically getting remodeled. We're getting more neurotransmitter or some change in that neuron, which is going to make it more functional, more responsive, more capable. And that might be one of the major mechanisms that are involved with how we learn. Here's a little bit of research that Eric Kandel did on that same topic. I'll get into that with you guys later. And this concept of what LTP is, the idea that these synaptic areas can change and become far more responsive. And here's just a little view just to remind you of what that is. So here's the initial activity at these two neurons in their synapse. And then when we have repeated stimulation on that same circuit, 1 week later, we notice that there's this postsynaptic side is broader. It has more receptors in it. We have more neurotransmitter over here. And so we get a much bigger response.